Hello everyone. So today we are going to discuss about the immunotherapeutics. In this seminar, I will cover the concept of immunity, types of immunotherapeutics, then active and passive immunotherapy, then immune checkpoint inhibitors, adaptive cell therapy, and their application in modern medicine. I would like to thank every resources which gives me the information about the immunotherapeutics. Before discussing the immunotherapeutics, let us understand the immunity. Immunity is the body's natural defense mechanism against the harmful pathogens like the bacteria, viruses, and other foreign particles and substance. The study of immunity is called the immunology. So, what we say that the study of immunity is called immunology. Then, when a antigen when a foreign substance we call as antigen, when an antigen introduced into the body, introduced in our body, so bodies produce antibodies against that particular antigen. So if our antibodies are able to eliminate the antigen, then we are free from that particular disease. But when antibodies are not enough or able to eradicate the antigen, we suffer from the particular disease. So when an antigen enters in the body, antibodies are produced to neutralize that particular antigen. If the immune response is effective, disease is prevented, and otherwise illness may occur. Then introduction of the immunotherapeutics. Immunotherapeutics are is an agent. So immunotherapeutics are agents either activate the immune responses or suppress the immune responses. They have two different applications. Either they activate that particular immune response or suppress the particular immune response to treat the particular disease. So these therapies are used in the conditions like cancer, then autoimmune disorders, then infectious disease, then which are further strengthened by the cutting edge treatments like the checkpoint inhibitors, immune checkpoint inhibitors, monoclonal antibodies, cancer vaccine, adaptive cell therapies, like that. So they works by enhancing the body's natural defense mechanism and provide more targeted treatment with fewer side effects and compared to the as compared to the conventional therapies. As compared to the conventional therapies, these immunotherapeutics have high application in treatment of the particular diseases. Then history of the immunotherapeutics. So there is a scientist which known as Cole. The name of the scientist is William Cole. What he do that, which is a New York surgeon, first noted that an infection after the surgery seems to help the some patients which are suffering from the cancer. So he observed that some cancer patients improved after developing bacterial infection. He developed a treatment known as Cole's toxin. Cole's toxins by treat the particular cancer by injecting the streptococcal organism into the cancer patient in order to causes the erysipelas, which is the red patches on the skin, and stimulate the immune system to stimulate the immune system. So, although his approaches was later replaced by radiation therapy, it laid the foundation for modern immunotherapeutics. So, today's immunotherapy therapy has become an important treatment option for several type of cancer. So, this is how the immunotherapeutics are developed, or the history behind that Cole's toxins. Then type of immunotherapeutics. So there are two different types of immunotherapeutics. So first is active immunotherapeutics and second is passive immunotherapeutics. So active immunotherapeutics stimulates the patient own immune system. That means the patient own immune system just uh, uh, are there, so they stimulate that particular immune system. Whereas in passive immunotherapeutics, uh, ready-made immune components such as antibodies are given. So here we see that adoptive cell therapies is there. So in the case of passive, the adoptive cells were transferred, or monoclonal bodies were transferred to the patient. While in active immunotherapy therapy, there is nothing like that to give the antibodies or something. But in active immunotherapeutics, we give the vaccines, or we can give the some immune check inhibitors or stimulatory cytokines to stimulate the immune system. This is the main difference between both of them. Then active immunotherapeutics, so first we move toward the active immunotherapeutics. So active immune therapeutics, which is work by stimulating the host immune system. So it stimulate the host immune system rather than uh, giving the patient the ready-made monoclonal antibodies or cell therapies. We only stimulate the immune system in the active immunotherapeutics. So or a specific responses we can stimulate to a disease or a pathogen, and is most commonly used in the cancer treatment. So it is commonly used in cancer treatment, and it's also being studied for neurodegenerative disorders like Parkinson disease, Alzheimer's disease, prion disease, then multiple sclerosis. Then active immunotherapy can produce both general as well as a specific immune responses, which is depend upon the therapeutic goal, or which is depend upon the different kind of therapies. So in non-specific active immunotherapy are there, and specific active immunotherapy are there. So in non-specific active immunotherapy, which generate a general immune system response using the cytokines and other molecules. So general immune system responses it generate. While in specific active immunotherapy, so the generation of cell mediated and antibody immune responses, which is focused on specific antigen expressed by the cancer cells, typically using the vaccine platform. So in active specific active immunotherapy, there is a specific antigens or specific immune responses which can be generated. But in non-specific active immunotherapy, there is generation of the general immune responses. Then in specific active immunotherapy, as we all know, it produces or stimulate the specific immune responses. So the first example of specific immunotherapy, specific active immunotherapeutics, are the cancer vaccines. So cancer therapeutic vaccines can be classified either cell based or antigen based. So, in the case of cell based, so in the case of cell based, the cell transfer is used in cell based vaccination. So, cell transfers are there in the cell based, but to elucidate the target anti tumor immunity. Some examples of the whole cell vaccines are adaptive T cell therapy, in uh, in which healthy T cells are transferred to a patient, and allogenic cancer cells. On the other hand, vaccine based on antigen intended to improve the ability of the body to defend itself by introducing the new antigen or different way of presenting the existing one. So, in specific immunotherapy, target particular antigen present on cancer cell. So cancer vaccines are major example. These vaccines can be cell-based or antigen-based. Cell-based vaccines use immune cells to generate anti-tumor immunity, while antigen-based vaccine introduce specific tumor antigens to stimulate the body immune system against the cancer cell. Then non-specific and active immunotherapy. So in case of non-specific active immunotherapeutics, so non-specific active immunotherapy stimulates the immune system in a general manner rather than targeting a specific antigen. As we all know, the aim is to enhance the body's overall immune responses. So it is mainly focused on stimulating the um, overall immune responses rather than focusing on the specific antigen or specific target. So the aim is to enhance the body's overall immune response so that it can eliminate the abnormal or cancerous cell. So cytokines are most commonly used approach in this case. So it is an immunomodulating agent which regulates the immune system responses and produces by various immune cells. So these agents include the following agents and markers. So there are cytokines, then chemokines, interleukins, and the example of this all. Then cytokines are the group of proteins that bring about communication between cell types involved in the immunity. So cytokines are small proteins that act as a messenger molecule between the immune cells. They regulate the immune as well as inflammatory responses. Important cytokines include the interleukins, interferons, tumor necrosis factors, as well as the chemokines. So this molecule helps coordinate the body's defense mechanism and essential for effective immune responses. So in specific, so cytokines are the group of proteins which bring the communication between the different cells which is involved in the immunity. They are low molecular weight glycoproteins which is produced by the lymphoid and non-lymphoid cells. During the course of immune responses it get produced. So cytokines may be regarded as soluble messenger molecule of immune system. They act as a short messengers between the cell and long range messenger by circulating in the blood. Then the later function is comparable to that of hormone or the and the term include interleukins is frequently used to represent the cytokines. There are more than dozens of interleukins are there, which is produced by different cells with a wide range of functions. The main function is of the cytokines is to amplify the immune responses as well as the inflammatory responses. So therapeutic use of cytokines is nothing but it is now possible to produce the cytokines in vitro. Some of the cytokines have potential application in the practice of medicines. For instance, the interleukins 2 is used in the cancer immunotherapy and in the treatment of immunodeficiency diseases. Then interleukin 2 also induce the proliferation and the differentiation of the T and B cells. Then beside increasing the cytotoxic capacity of the natural killer cells, a group of cytokines, namely interferons, can combat viral infection by inhibiting their replication. So it inhibits their replication with the interferons. Then passive immunotherapeutics. So we see the active immunotherapeutics. Now we are moving toward the passive immunotherapeutics. So passive immunotherapy involves the administration of performed antibodies to the patient. So it does not depends upon the stimulating the immune responses, but it is depend upon the administration of the antibodies like such as immunoglobulins to prevent or to target or kill pathogens or a cancer cells. It is designed to draw support from the foreign antibodies that are donated by from a person, extracted from animals or made in the laboratory to elicit an immune responses instead of relying on the innate immunity system to fight disease. It has long history from 18th century for treating infectious disease and is now used in the cancer treatment widely. So passive immunotherapy involves the administration of perform antibody to the patient. Instead of stimulating the patient immune system, it provides immediate protection using the antibody obtained from the humans, animals, or laboratory production. So this approach has been used for many years in treating infectious disease as well as the cancer. Moving forward,